Chapter 6, Section 5, Regulatory Enzymes. Excuse me. Regulatory enzymes. These are going to be enzymes that increase or decrease in response to certain signals. Okay. Up to now, any enzyme we've talked about, it was just go, go, go. As long as there's substrate present, they're going to work. Well, sometimes you want to be able to fine tune whether or not, turn it up, turn it down, the activity of an enzyme. Okay, based on the current at that instant cell's needs. You can have what are referred to as allosteric enzymes. Okay, these are enzymes that function through reversible non-covalent bonding. Okay, you'll have modulators or what they call allosteric effectors. Okay, here's where cofactors or other organic compounds or small things can, associate or disassociate with the enzyme, and that association could cause the conformational change required to make the enzyme work, or the association could cause the conformational change of the enzyme that alters the shape enough that it doesn't function. There's also going to be a form of, covalent modification. Okay, you're going to covalent bond. Okay, this is, a reversible. You're going to get something bound to it. Maybe it's a phosphate group that gets bound to the enzyme. Okay, you put on a functional group like that onto an amino acid, you're going to change how that amino acid reacts to the other amino acids around it, changing phi-psi angles, and you could induce some change in the overall shape of the protein. Make it the correct shape, thus get a function, or change the shape and lose a function. Others going to have some form of proteolytic cleavage. After translation, that polypeptide may fold correctly. You get the correct secondary, tertiary, but there's a little too much on the tertiary, so you can't get the correct quaternary until another enzyme comes and cuts off some excess, and then that correct tertiary now becomes the correct quaternary. You have an active enzyme. Allosteric enzymes undergo conformational changes depending upon what does or does not bind there. You're going to have two domains, two parts. Here the artist has drawn it that the two parts are of equal size and whatnot. Doesn't always work that way. This regulatory domain could be much, much, much smaller than the catalytic domain. What's important, though, is this regulatory domain is going to bind some form of modulator. Here, this modulator, when it binds to the regulatory, it changes the conformation of the catalytic domain. The catalytic domain now has the correct shape, active site, so that the substrate can fit in. We refer to this as heterotropic modulation. This modulator is something other than the substrate. A homotropic, if the substrate can fit into the active site enough, there's enough interactions, it will cause the overall conformational change, kind of like a shift and transition state, as we've seen in previous sections. You have a regulatory enzyme known as aspartate transcarbamylase, ATCase. Takes the formation of the carbonyl carbamyl aspartate. Okay, this is part of the metabolic pathway that gives you the pyrimidines. Carbonyl phosphate and aspartate. You cut, you paste, remove this phosphate group using ATCase, and you get the beginning of the pyrimidine ring. What's happening here is that CTP cytosine triphosphate interacts with the enzyme, and it will negatively regulate what we just talked about on the previous. On the previous one, remember, you take the carboamyl blah blah blah, turn it into the beginning of the pyrimidine ring. You used ATP. So this right here, we're using CTP, which is cytosine triphosphate, similar but not really to adenine triphosphate. CTP binds, keeps everything in an inactive state. Remove the CTP, you now have an enzyme that is in the active R state. Need to turn this off? Well, add CTP to the mix. You hold it in an inactive T state. These allosteric enzymes, when you go and you try to plot them using the Michaelis-Menten behavior that we saw before, well, when you plot out V naught versus the concentration of S, you don't get a linear line like we saw before. You're going to get some form of a sigmoid saturation curve. Not that hyperbolic curve that you take the linear line to get the V0, remember? So instead of a nice hyperbolic that we saw before, you see more of this sigmoid, not quite approaching Vmax. So small increases here give us big curves. So instead of this voop, you get this more gentle curve up, and then shift over. More of this really stretched out S as opposed to the hyperbolic, which goes almost up and over. Because if you looked at here, the V naught of this allosteric, the V naught would be taking off over in this direction, as opposed to here, which would be going straight up. For heterotropic allosteric enzymes, we see that there's a difference between activators and inhibitors. Enzyme alone in black. As you increase the activators, you actually increase the kinetic rate. As you add in inhibitors, you get this shift where it takes more and more substrate before you're going to hit Km, or the half Vmax. For some of these, you can have a modulation where Vmax, when the enzyme is just maxed out, saturated. So our half Vmax, our Km, doesn't change. The Vmax just gets lowered and lowered and lowered. Because if you look at all of these, they are all pretty much in line wherever you see Vmax. And then that reversible covalent bonding. Hey, look, ATP. You can use phosphorylation, adenylation. Instead of just the third phosphate, you're going to clip off. Somebody in the lecture had asked once, well, does it always just clip off the third phosphate? No, here you're clipping off, the second and the third. And then you're adding the adenine to the enzyme. Acetylation, you're adding an acetyl group. What's also referred to as a myristylation. You're adding a myristyl group. ubiquitination. Ubiquitination, we've talked about this before. If a protein can't fold correctly, there are enzymes in the cytoplasm that will go through and add ubiquitin groups. Ubiquitin group is a big red flag to other enzymes to come and digest this away. Maybe it's adding in an AD ribosal. Go through and you add in this, modified adenine. You look at all this, instead of this single sugar, you now add in this double sugar thing. And then there's also just a plain old methylation. What do each of these do? Well, they change the polypeptide at the secondary level. Change the secondary, change the tertiary, change the quaternary. Many of these, the phosphate here, the adenine, the acetylation, ubiquitination, methylation, these can be added, these can be easily removed. Honestly, all of these are reversible, they can be removed, but we see mostly in when I've done research on cellular signaling, MAP kinase pathways and things like that, you're going to see phosphorylation, methylation, and lesser acetylation in those signaling enzymes. Ubiquitination is, for other signals required. That phosphoryl group from ATP is going to be added by what is known as a protein kinase. Okay, it's going to do what is called a substrate level phosphorylation. It's going to strip off that third phosphate, and depending upon what type of kinase it is, a serine protein kinase, a serine, tyrosine, histidine, whatever kind it is, it's going to go for certain serines, thyreins, tyrosines, and histidine in a specific sequence. And then it will add a phosphate group to it that has been stripped off of ATP. Anytime you do stuff like this, to make it reversible, you have to have something that pulls it off. That's the job of a phosphatase. Phosphatases are an enzyme. They go and they strip off phosphates off of polypeptides, proteins. So this phosphorylation, phosphorylase kinase takes ATP and strips off that terminal phosphate, adds it to phosphorylase. The kinase ends in ase. Kinase means it adds a phosphate group. And generally what prefaces that tells you the target, phosphorylase. Phosphorylase A is phosphorylated and thus active. To inactivate it, you take a phosphatase, phosphoprotein phosphatase, strips off the phosphates, adds the hydroxyls back. By adding those phosphate groups, as you see here, boom, boom, what is occurring is that by adding that phosphate group, you are changing the overall tertiary or secondary structure. Change the secondary, change the tertiary, change the quaternary. You go from a T state to an R state, which is now ready to interact with substrate. T state, unphosphorylated, not so much. Now, even though it's called a serine, kinase, serine protein kinase, or a tyrosine protein kinase, it's not every tyrosine, it's not every serine. It's only ones within certain motifs, okay? Motifs, or you'll see it also referred to as consensus sequences. Protein kinase A looks for X means anything, I forget what R and K is, anything, boom. There's the target. Next amino acids on. Every one of these kinases has a specific sequence that it has to look for. The variability is going to be whenever you see the little x's. That just means anything can be there. Now, to be activated, and have multiple phosphorylation sites. Means that for certain proteins that need to be activated, well, different kinases, not just one kinase can activate it. Again, you take that glycogen synthase. You have multiples, as you see here, that can activate it. Turn it off. Turn it on. Some of them have greater sensitivity than others. But if you'll notice, no two have exactly the same sites. Different kinases, different sites. Talked about the idea of a zymogen. Okay, this is a long polypeptide that's made during translation. It cannot get into its correct shape. So you look here at chymotrypsin. Chymotrypsinogen. This is the inactive form. Trypsin, chymotrypsin. So first you have the inactive form. Trypsin comes and cuts. Notice how it cleaves here between the 15th and 16th, this arginine isoleucine. This pi chymotrypsin is now an active form, and it can do what is referred to as autolysis. It can cut self. So certain amino acids, 14 and 15 and 147 and 148, get removed. You now have alpha chymotrypsin. All of this will fold correctly, and you get the correct quaternary form. Proproteins, proenzymes, these are just precursors. Things that need to be just trimmed up, cleaved, have an end cut off. Trypsinogen to the active trypsin. The interesting thing about having enzymes, as we've seen here, is you can have control. Is it going to be active, inactive? But again, these enzymes only do one thing. They cut, they paste, they twist, they tweak. Okay, they can't take a substrate and make a final product, unless it's a one-step thing, and rarely is it a one-step thing. So we put a series of enzymes together, a pathway. By having a pathway, each enzyme, each step in that pathway gives us another chance to regulate. Does this actually have to happen? Yes, no. Move on to the next step. Does this actually have to happen? Yes, no. Move on, move on. Blood clotting. Great example of this. Blood clots are an aggregate of cell fragments, platelets. They go and they start aggregating, they get activated, they start binding together, proteins bind them together. And as they're aggregating, they're trapping all other blood cells there, red blood cells, white blood cells, any bacteria or whatever that may be there. They will be stabilized by these fibers you see here. Fibrin. Fibrin, as you see here, holding all this together in a meshwork that will dry out and become what we think of as a blood clot or a scab, starts off as fibrinogen. Fibrinogen doesn't entangle like this, okay? It doesn't entangle like we see this lattice work here. Free floating in the blood. But a series of reactions will take this and cause it to start to look like this, which will lead to what we see here up in A. Collagen gets exposed to blood. Collagen should never see all the blood's components. Okay, collagen may see, fluid, may see some of the proteins and all, but it's not going to see everything there. This exposure triggers the release of signal molecules, thromboxanes, which activate platelets, cause the platelets now to start going in fibrinogen. Fibrinogen converted via proteolytic removal. Okay, it goes through, starts removing. Thrombin is our enzyme of choice here. Goes at the serines, certain serines, and catalytically removes so that fibrinogen can become fibrin. Transglutaminase enzyme, also known as factor 13A, aids in the cross-linking of fibrin. So you now start to get the meshwork formed. So you start looking at all of this going on here. The arrows, boom, 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 boom. These are the steps. Other things feed back and cause more to happen. That's the green. Wherever you see red, that's a regulatory. Stop. Don't do. Because think about it. If you didn't have all these regulatory stops, don't do, you would be throwing clots left and right all day long. Other times you're going to have multiple enzymes, multiple things going on. And if you have multiple things going on, you're going to have multiple control pathways, multiple control mechanisms. Okay, say the initial substrate is lacking in the cell. You don't want to get rid of all the enzymes, so you want to turn everything down. Let that substrate level build up to a certain threshold and then turn everything back on. Work down the substrate, build up the product. Hey, guess what? We have more than enough product. Don't care how much substrate's there. Turn everything off. Let the product level go down again, and then turn the pathway back on.